All right, let's get started. I have the great pleasure of introducing Will and his dissertation defense. Will came to us from UC Boulder, where he was a double major and was already working on methods for using machine learning and computer vision for extracting traits from herbarium sheets. This is work that he's continued to do here for the last several years. Will is extremely efficient and productive. So he has written several programs. So Leaf Machine 2, Voucher Vision, Field Prism, and probably other things that he's reluctant to tell me about. At least Field Prism and Voucher Vision were things where I think you had probably already written half of them. And then it wanted to have a meeting with me to say, do you mind if I do this? I've already done it. And it's not instead of something else, but in addition to the work that he's already been doing. He's published 11 papers, either as first author or collaborator. 9 of those, I think, while he was here at University of Michigan. They're in places like New Phytologist, our like flagship fanciest plant journal, American Journal of Botany, Application in Plant Sciences, and other things I can't remember right now. I suspect that we will be seeing some really fancy publications coming out of his work soon. What else? I guess in a word, Will is extraordinary. All of this stuff, all of his success and all of this work is not luck. It's just hard work. He is on Discord till 3 a.m. He's answering emails at all time. Thousands and thousands of lines of code. And I think it's hard to really communicate exactly how unusual it is to have a graduate student that has already had such a large impact on a field. So one of the publications that Will is on is on establishing standards in machine learning and AI for the plant sciences and for plant journals. As a grad student, that's pretty amazing. But then his software is already being used by dozens of herbaria across the world, including probably the largest and most important herbarium in the world, Kew Gardens. The University of Michigan herbarium is size does not matter. It is extremely important. But Kew Gardens, New York Botanic Garden, all of these places rely on Will software to do work, which is pretty amazing. Anyway, I don't want to take up any more of your time, but look forward to this talk. Should be great.
Thank you for that introduction, Stephen. And thank you all for being here with me today. It means a lot to see all of you. So it's amazing how this grad school journey felt like it would never end, but here we are. On your way into this building today, or when you stepped outside this morning, you passed by thousands of tiny biological machines that were simultaneously harvesting sunlight, processing carbon dioxide, and producing the oxygen that we're breathing right now. And most people walk by them without giving them a second thought. But I'm the odd one out. For the last 10 years, I've been completely obsessed with leaves. And by the time we're done here today, I hope that you'll never look at a leaf the same way again, for better or worse. For my talk, we'll begin with some background information and the motivation for my research, and then we'll look more closely at each of my chapters. Chapters 1, 2, and 3 of my dissertation focus on methods for extracting unprecedented amounts of plant trait data from digitized herbarium specimen images or from field images. Chapters 1 through 3 have already been published, so I'll be not spending very much time on chapters 2 and 3. If you want to find out more about them, though, you can go to my CV webpage and find links to all of my various websites for all of my projects. And then in chapter 4, I apply all of these methods to look at leaf shape variation and diversity across 12 really interesting plant families. And then finally, we'll end with a discussion about future directions and what I have planned next. So to start very broadly and get us all on the same page, this is a phylogeny of Verdiplantae, or green plants. And in this phylogeny, near the bottom, we have things like algae, And as we work towards the top, we start to get more complex plants, eventually angiosperms or flowering plants there at the top. And one important thing to note about green plants is that they are not evenly distributed taxonomically. For example, there are roughly 1,000 extant living gymnosperm species. So that's things like pine trees. But there are well over 250,000 flowering plant species, and perhaps as many as 350,000, depending on how we define them. The impact of angiosperm species on biodiversity is truly astonishing. Darwin famously remarked that the origin of angiosperm diversity is an abominable mystery, and that's still largely true. And today we're going to be focusing on angiosperms, and specifically the astonishing diversity of angiosperm leaves. Studying leaves dates back to the Victorian period. Unsurprisingly, Darwin was a fervent botanist, and more of his time was spent studying plants than any other system. The incredible diversity of plants and their representation in the fossil record spurred much of Darwin's research, and he rigorously tested his theories by breeding and crossing plants in his gardens. Another early botanist, Johann Wolfgang von Goethe, observed that leaves were essentially building blocks for plants, and that many plant features are derived from leaves. Leaves come in all shapes and sizes, so it's helpful to know a few of the terms that describe their architecture. So on the right-hand side, you'll see an example of serrate and toothed margins that you might find on an elm tree. Above that is a smooth entire margin. You might find that on a rhododendron. And then on the left side, we have an example of a lobe and a sinus, which are common on oak trees. And then below that, we have an example of a leaflet, which is part of a compound leaf that we'll discuss in a moment. And then at the bottom, we have a petiole, which is attached to the stem. So to complicate matters, each of these photos shows only one leaf. On the left is what most of us would think and visualize as a single leaf. That's a simple leaf from a white mulberry. In the middle is an example of a pinnately compound leaf from smooth sumac. And on the right is a bipinnately compound leaf from Kentucky coffee tree. And the green arrows highlight a leaflet as part of the larger compound leaf. Those are fairly typical leaves, but these are examples of some highly modified leaves that show just how crazy angiosperms are. Each of these leaves has been co-opted for another purpose. Sarracenia and Nepenthes are pitcher plants, and their leaves have been modified to trap insects. Cacti modified their leaves into spines for protection and shading. And then the genus Gloriosa has modified leaves that act as tendrils so it can help the plant climb. So that's just a small taste of some of the extreme variation in leaf form and function that we see across flowering plants. But in botany, flowers tend to get all of the attention. But in case you didn't know, flowers are also derived from leaves. So if you ask me, leaves are quite literally the bee's knees of all life on Earth. Flowers are simply glorified leaves in disguise, leaves that co-evolved with bees who in turn facilitated the radiation of 250,000 flowering plant species, which in one way or another provide most of the calories that humans consume. And with our stomachs full, we had time to be curious about the plants that fed us, leading to generations of talented botanists, and finally to you sitting in this room today listening to me rant. Since leaves are the most important biological structure on Earth, you'd think that we would know almost everything about them by now. But there's a plot twist. Leaves are incredibly diverse and complex structures that exhibit variation at many different scales. There's a tremendous amount of leaf trait variation even within a single species. Individual plants often display a wide variety of leaf traits. For example, oaks tend to have highly variable leaves even within the same individual tree. Their shapes are often dependent on their location in the canopy, the timing of bud burst, and how much light they're exposed to. So you can imagine that among oaks within a population in a forest, there's even more variation. And some oaks have huge ranges, like the bur oak, which occupies much of the North American continent and displays quite different leaf shapes across their range. They tend to show more full, filled out leaves in the northern regions of Canada, while they tend to appear more dissected in their southern regions, like this leaf from Texas. All of this needs to be accounted for if we want to quantify variation for the species as a whole. Botanists and plant systematists love to look for similarities and differences between and among taxa. These diagrams are from Leo Hickey and Jack Wolf's 1975 paper, where they group taxa by leaf architectural features to identify phylogenetically informative patterns of leaf architecture among angiosperms. They systematically cataloged features like leaf margin type, venation patterns, and overall leaf organization, which created a comprehensive morphological framework to reveal how leaf architecture could inform our understanding of angiosperms and their evolution. James Doyle revisited their paper in 2007, where he used molecular data to revise some of their original proposed relationships. He demonstrated that when leaf architectural characters were mapped onto molecular phylogenies, many of the morphological patterns were actually more consistent with the newer molecular trees than with older morphology-based classification systems. That allowed him to distinguish between evolutionary relationships and misleading trait convergences that had confused earlier systematists. Doyle's work revealed the power of combining molecular phylogenies with morphological data to better understand leaf evolution, but it also raised new questions about the drivers of leaf diversity. Beyond phylogenetic relationships, leaf shape has long been linked to climate. Over a century ago, in 1916, Bailey and Sinnott observed that only 10 to 14% of temperate woody plants have entire smooth leaf margins compared to the majority of tropical species. So today I'm going to be using the word entire and non-entire a lot, so you'll see this key in the bottom right-hand corner, just so you can visualize what I'm talking about. So visually, botanists, including Bailey and Sinnott, would observe leaves that look something like this in northern, colder, temperate regions, where you see a lot of dissected leaves, a lot of leaf teeth. And they would have observed leaves that look like this in the tropics. Most of the leaves tend to be uniform. They have smoother margins, and they're generally sort of oval shaped. But the reality is always more complicated. Some taxonomic groups are overrepresented in certain geographic regions, which means that it's a good idea to try to account for spatial dynamics and phylogenetic relatedness. So if we want to look at large-scale patterns between biomes and across continents, we need to add one more level of variation to our diagram, and that is interspecific variation, the difference between species. For an example of interspecific variation, we can look at this phylogeny from Givnish and Kriebel, 2017, which maps the presence or absence of leaf teeth broadly across angiosperms. So here, black dots represent the presence of leaf teeth, and white dots are smooth entire margins. If we focus on just the order Dipsacales here in red, we can see why the scale of our analysis matters quite a bit. The red is a zoomed in portion from the order level phylogeny on the previous slide, and on the left is a phylogeny for the genus Viburnum, which is inside the order Dipsacales. So from the order level phylogeny, we can only identify at most 2 transitions from entire to non-entire, but at the species level, we're able to document maybe 8 to 14 changes. So ideally, we want to build data sets that allow us to have species level resolution, but ideally, we want to see patterns more broadly across angiosperms. So in these 2 studies, they were looking for the presence or absence of leaf teeth. And for some context, there are a handful of hypotheses for what drives the evolution of leaf teeth and more complex leaf shapes in general. The first is the support and supply hypothesis, which suggests that leaf teeth are simply a structural consequence of strong veins that are required to support the thin leaves that we tend to find in colder climates. There's the bud packing hypothesis, which posits that toothed margins are a side effect of the dense and intricate packing of young leaves within protective overwinter buds. Other studies rejected both of these explanations, suggesting instead that leaf teeth provide a direct advantage by boosting photosynthesis early in the short growing season of colder regions. Leaf teeth have also been suggested to reduce herbivory from insects and animals. But we also need to account for phylogenetic relationships because some lineages can become developmentally constrained. And that means that tooth margins might persist as an inherited trait rather than a direct adaptation. particularly if selective pressures are weak and the feature is carried along by genetic drift or pleiotropy. So the current utility of a feature is not always a good explanation for why this trait exists right now. For example, leaf teeth might have evolved as a defense against herbivory at the blue dot, but leaf teeth could also confer an advantage for cold tolerance, which would allow descendants to occupy colder regions. And that would be an example of next adaptation. We could also choose to focus more on the ecology of the plants, perhaps at a global scale. Write et al. In 2017 provided a quantitative explanation for latitudinal gradients in leaf size by demonstrating that large leaves predominate in wet, hot, sunny environments, while smaller leaves tend to occur in arid regions. Pepe et al. In 2011 also conducted a global analysis of leaves, and they showed that in colder climates, the leaves tend to be dissected or toothed, and that leaf habit, water availability, and phylogenetic history were all significant drivers of those patterns. So this striking pattern of latitudinal gradient has been observed repeatedly worldwide. So where do we go next? Most leaf-centric research has fallen into one of two categories. They've either tended to be clade-focused or focusing on functional traits, perhaps at a global scale. And these have been very fruitful strategies, but the only reason that we have not been able to merge these two together is a lack of data. So in chapter 4 of my dissertation, I present a preview of future macro-scale evolutionary biology and ecological research, where we can use machine learning, computer vision, AI tools, and clever data processing pipelines to methodically extract granular data from natural history collections with replication at the individual level across populations and biomes through time for hundreds of thousands of species. We can begin to chip away at really big questions. So are climate leaf trait relationships truly adaptive responses to environmental pressures, or could they arise from neutral evolutionary processes like genetic drift? And critically, are these patterns universal across all plant lineages, or might they be lineage specific? The amount of leaf trait data available to address biological questions like these has been steadily increasing, but is still relatively small. The largest data set to date is from 2016. They mined 6 traits for about 1 million leaves. In order to bring substantially more data into this conversation, I have relied heavily on digitized herbarium specimens and machine learning tools. The humble herbarium is truly a magical place, and it's one of my favorite places in the whole world. Herbarium vouchers are widely used in biological research. They're useful for their historical record. We can obtain genetic information from them. We can monitor the spread of invasive species, and they can be used to inform conservation status. These are some artistic examples of herbarium specimen vouchers. On the left is coffee collected in 1780. The middle shows tea varieties collected in 1841. And on the right is liquid ambar strassiflua, which is sweet gum and one of my favorite trees. While vouchers can be used for many purposes, I'm primarily interested in extracting quantitative trait data from these beautiful collections. And to do that, I've built a lot of different machine learning tools to primarily address these 3 bottlenecks. We want to automate labor-intensive tasks to do things like segmenting out individual leaves from the images or identifying specific leaf features. We want to mobilize large data sets, make them discoverable, and make them available to more researchers. And we also want to harness the power of computational morphometrics to generate massive trait data sets to help us begin to address these large biological questions. So that brings us to Leaf Machine 2. My goal was to make generalizable algorithms that could extract quantitative trait data from most taxa, from most herbaria. I created a modular machine learning framework based on a delegation strategy where we essentially divide a complicated task into smaller solvable components so that we could more effectively extract research-grade measurements. Leaf Machine 2 leverages an ensemble of 7 different machine learning algorithms to extract lots of different information from our images, and we can summarize all of that information in the image on the right. This diagram outlines the modularity of Leaf Machine 2. Here, each box represents a distinct machine learning algorithm. Green are object detection algorithms, which place bounding boxes around important features. Cyan are large language models that deal with text in the images. Gray are scene detection algorithms. Orange are key point detection. And white is instance segmentation, which is responsible for actually finding and isolating the leaves in the images. The output from all of these models is then aggregated together, which produces a huge amount of derived trait data and useful archival metadata. Over the years, I've had to generate a huge amount of training data to make all of this possible. So this involves having a person manually draw bounding boxes around all of the things we're interested in and manually tracing leaves. That provides us with the training data necessary to make machine learning models. And I want to thank all of these wonderful folks who've helped generate all of this data. It took well over 2,000 hours of painstaking labor, but as a result, we've been able to do really cool things. We were able to annotate more than 500,000 objects for our plant component detector, 200,000 objects for the archival component detector. We placed 250,000 landmarks on individual leaves, and we manually traced more than 30,000 leaves in a bunch of images, which gave us more than 1 million annotations, which is by far the largest set of training data for any natural history use case. This is the basic outline of Leaf Machine 2. The first thing to notice is that the archival component detector and plant component detector drive the entire process. That's number 2 and number 6 in this diagram. They are responsible for creating bite-sized pieces that we can then analyze with component-specific analyses. So now let's follow this specimen as we process it with Leaf Machine 2. We start by identifying 11 different plant components, including ideal leaves, partial leaves, buds, fruits, flowers, roots. The bounding boxes isolate each of these components so we can work with them individually. Once we've isolated something like an ideal leaf, we can send them to be processed by the segmentation and landmarking algorithms, which looks something like this. The green masks are used to calculate morphological properties like leaf area, perimeter, and other shape-based measurements. And since this process is modular, we don't have to waste time dealing with partial leaves or something that we don't actually want to get a measurement from. We also detect 11 different landmarks to generate another set of measurements, including tracing the petiole, tracing the laminar midvein. And stepping back, we can overlay all of this onto our final quality control summary image. I wanted to highlight some of the segmentation abilities of Leaf Machine 2. We can produce high-quality masks even for complicated and difficult scenarios. We trained our segmentation algorithm to specifically ignore mounting tape, which is on the bottom row. Many herbaria use mounting tape to attach the specimen to the piece of paper, and quite often, every single leaf on the paper is bisected by mounting tape. And before Leaf Machine 2, you'd have had to manually, reconstruct the entire leaf, but we solved that with Leaf Machine 2. You'll also notice that Leaf Machine 2 segments individual petioles, so those are the blue areas and orange areas in some of these images, which we just used as part of a workflow to automate the estimation of leaf mass per area. We showed that an automated computer vision pipeline can reliably extract leaf area and petiole width from herbarium specimens at an unprecedented phylogenetic and geographic scale, which helps fill global trait data gaps. Those are some of the things we can do with plants, but now we need to take a closer look at the archival components in these images. We can identify 9 different archival components, and that's things like the label text, the barcodes, the color correction cards. But something that I've spent way too much time working on is how to handle rulers in all of these images. I have a love-hate relationship with rulers in herbarium images. On the one hand, they make it possible for me to obtain trait data from all of these images. But on the other hand, I've had to spend an insane amount of time developing algorithms to identify tick marks in all of these different rulers. And this is just a summary of the variety of rulers that we've come across. After some data crunching, I was able to make recommendations for future digitization efforts, showing that these 2 rulers should be used at all costs, if possible. They perform consistently, they're machine readable, and it just makes life a lot easier. They also contain multiple unit markers, which allow us to cross-validate our pixel to metric conversion factors so that we can autonomously validate that the measurement we're getting is real. One of the many side projects of my PhD was making 3D printed rulers, and I'm still way too excited about this. I have them with me up here if you want to see them after. Yeah, so all you need to make them for yourself is a $250,000 Stratasys J850 multi-material 3D printer, and you too can have 3 rulers. So that covers Leaf Machine 2, and we'll move pretty quickly through chapters 2 and 3. While Leaf Machine 2 is built for herbarium vouchers, oftentimes we also want to collect data from images in the field. For that, I developed Field Prism as a way to bring an element of standardization that we have in herbaria into the field. Field Prism is a system of simple photogrammetric templates and QR codes that are useful for organizing and standardizing images in the field. When you're in the field, it's often difficult to photograph specimens in a way that's conducive for obtaining accurate measurements. So this software leverages photogrammetric templates to correct for image distortion. You can use any camera you want. You can use your smartphone, or you can build one of these imaging stations. I have one sitting in front if you want to take a look at that. The one on the right is also wearable, and I promise you'll be the coolest person in the forest if you're walking around wearing that. So the software corrects image distortion, as we can see in this before and after. And it also calculates pixel to metric conversion factors, just like we do in Leaf Machine 2. One exciting use case for this is the ability to create time series data sets. So you could do something like track leaf development. These are just proof of concepts, but you could imagine that you could do some really cool projects with something like this. After that brief detour, we're moving on to chapter 3. And while Leaf Machine and Field Prism are designed to handle plant components, I've also been building software to extract information from the labels that accompany herbarium specimens. Voucher Vision is an AI-powered toolkit for reading, parsing, and formatting label text into standardized data structures. So I leverage quite a few different large language models to accomplish this task. Large language models encompass a wide variety of products that you might be familiar with, including Google's Gemini or OpenAI's ChatGPT. And although those might be fun to interact with as a chatbot, we can actually use them to do a lot of really useful work. They're incredibly powerful at reading text in images. And in my case, they're incredibly powerful at translating text in an image into a standardized format and rearranging it to fit whatever purpose we need. In museum collections, in order for the specimen to be entered into a database, for it to become searchable, someone has to manually transcribe all of the text in the labels for every herbarium specimen. And as you can imagine, that's a very laborious and expensive process. These are just a handful of examples of something you might see in an herbarium collection. And we can see that there's a huge variety of readability in these images, mostly dependent on how old the specimen is. Modern labels tend to be typed out. They're usually organized. And the older you get, the more you run into very hard to read cursive. You start to see taxonomic revisions, and quite often the labels even contain conflicting information. For the current manual transcription process, where a curator parses and types out relevant content into specific fields and then enters it into a database, a curator might manage 15 to 20 images per hour if everything is going smoothly. And the most time-consuming part of this process is if the person has to query an external data set to validate information, like perform a Google search to verify a geographic location, or if they have to reference some other specimen that's already in the database. With older labels, a curator might only manage 10 to 15 specimens per hour, but it could take even longer than that. The oldest specimens could be hundreds of years old. So you can imagine that it could be challenging to rectify things like geography. Geography has changed a lot in the last 200 years. To put all of this into context, only about 48 million specimens have been databased globally, but there are well over 300 million specimens still sitting in the stacks that cannot be easily accessed or searched. And a significant portion of those have not even been accessioned. So we have no idea what's in the boxes in a lot of collections. So if we do some napkin math, it'll take decades for us to work through that backlog. So any tool that can help us increase the efficiency and reduce the cost of transcription is a huge benefit to many institutions around the world. And critically, adding these records to online data portals will make them findable and accessible, greatly expanding our understanding of global biodiversity and providing more insight into our changing world. I want to provide a little bit of an overview of how voucher vision works. We start with the full specimen image. We then use a piece of the Leaf Machine 2 pipeline to create a label collage. This label collage is nice because it lets the large language model ignore all of the complicated plant material. From there, we extract unformatted OCR text from the image. Once we have the text, we provide instructions to the large language model. We create an image-specific prompt that guides the large language model through the process of converting the messy text into a consistent JSON dictionary. And if that JSON dictionary passes our quality checks, we can add it as a row in our spreadsheet, and it can be reviewed by a human before we finally upload it into our database of record. And once it's in the database of record, the public has access. So instead of 15 to 20 images per hour, now we can transcribe up to 6,000 images per hour. And the AI transcriptions still have to be reviewed by a person, and that adds time. But having this preliminary transcription data is still incredibly valuable for internal research. So it's still a very, very important thing. In the past few months, I've developed an API for voucher vision, which makes it pretty easy to use. You can go to this QR code. It'll take you to a website, and you can just play around with it. The API also has an associated Python library, so you can add it to any work project you'd like. This project is taking on a life of its own, which I think is fantastic. I want to acknowledge the wonderful folks who've helped me get voucher vision to where it is, including James Mickley at Oregon State University, who's been a huge help testing and developing voucher vision and making it usable by the larger community. I want to thank Andrew Hipp for all of the fun conversations we've had about voucher vision. And finally, all of the folks at Royal Botanic Gardens Kew, who are also helping take voucher vision from just a toy that I've been making to something that's actually usable by the larger community. With the help of all of my collaborators, we are working to integrate voucher vision directly into content management portals, including Symbiota and Specify in the United States, and the Disco platform, which is being developed for the European Union. And so far, the API has been used to transcribe more than 100,000 specimens, and we're just getting started. So, moving right along to chapter 4. So at this point, you might be thinking that I'm getting a PhD in computer science or leaf wrangling, but I promise there's plenty of biology here too. So let's get back to some of the things that I mentioned in the introduction, and that is the association of leaf shapes with climate. You'll recall that botanists have long observed a latitudinal gradient in size and shape, where smooth, entire leaves tend to show up in warmer, wetter regions, and non-entire leaves tend to be found in cooler, drier regions. So for chapter 4, some of my guiding questions are, do leaves get bigger or smaller in hotter environments? What about wetter environments? And then to focus on shape, do leaves get more or less complex in hotter environments? Do leaves get more or less complex in wetter environments? We saw this chart earlier, and now we can see how the Leaf Machine 2 data extraction compares. The previous study mined 6 traits from all available public data sets. And that compares to chapter 4 of my dissertation, where we can extract more than 30 different traits from basically any herbarium specimen we want. And in this case, the first large-scale use of Leaf Machine 2, I extracted 4.6 million leaves from about 500,000 herbarium specimens, spanning about 7,000 species. So what does all of this data look like? Well, for each full herbarium specimen, Leaf Machine identifies and isolates all of the complete leaves, resulting in the white segmentation masks you see on the right. And with the leaf outline mask, we can measure many different morphological properties. There are three traditional ways to characterize leaf shape. Traditional morphometrics rely on basic measurements that someone could do with a ruler or grid paper. That's things like leaf area, perimeter, aspect ratio. And these measurements are still very important, but they can't capture the complete complexity of a leaf shape. So an alternative would be something like geometric morphometrics, which are computationally derived and rely on homologous landmarks being placed on the leaf. This figure is a geometric morphometrics approach from the program MASS, where leaf outline is essentially characterized as an elliptical Fourier descriptor, where we fit a curve around the leaf shape. And then we could use something like Procrustes analyses to align all of these shapes and perhaps compute a mean leaf shape. Whether we use landmarks or elliptic Fourier descriptors, we run into a big problem. Landmarks require homologous structures, which we don't necessarily have if our goal is to work with all angiosperms. And comparing elliptic Fourier descriptors is also not necessarily possible at this scale. So we need a method that would allow us to compare disparate leaf shapes in a unified morphospace. For all 4.6 million leaves, I measured dozens of shape properties, but for looking at climate associations in particular, we're going to focus on leaf area, which is well represented in literature. But I'm also going to introduce a new way of classifying leaf margin that can actually scale to work with our large data set. This begins with a method called Euler characteristic transform, which I then distill into a binary or categorical representation using something called a geomorphon classification. Euler characteristic transform is a mathematical tool that captures the signature of any shape by systematically scanning it from different directions. So for leaf shapes, we're essentially drawing a line across the leaf. And as we move down the line, we're counting how many times we enter or exit the interior of the leaf. We repeat this process from many different angles, gradually building up these red waveform images where higher intensities indicate a more complex leaf shape. And from that, we can create unique mathematical fingerprints that can distinguish between different leaf shapes. Each of the red waveform matrices on the previous slide contained thousands of features. We could apply dimensionality reduction techniques to stratify and group these different leaf shapes. This is an example of a fate plot of 25,000 leaves and 51 species. And we can already see a lot of separation happening in this latent space. ACT matrices are very highly dimensional. Each leaf produces a matrix of over 16,000 values. So to make this computationally tractable for 4.6 million leaves, I developed a geomorphon classification approach that treats each ECT matrix like a topographic landscape. So we can identify ridges and plateaus in this landscape, and those correspond to different types of leaf margins. Ridges identify minor marginal features like leaf teeth, and plateaus identify major marginal features like lobes. Plot A in the top left corner is an example of a rhododendron leaf with an entire margin, but it has one small defect, which is helpful for getting our bearing and interpreting these images. So you can see what happens when we trace that one small defect through the red image. Geomorphon classification allows us to isolate the pixels in the ECT image that correspond to minor and major marginal features. So here, the green line moving through the image indicates that that small defect is identified as a minor marginal feature. And since the maximum intensity of each ECT matrix is indeterminate, geomorphon analysis is helpful because it allows us to compress all of that variation into just two classes, which are the blue and the green areas, again, corresponding to minor and major marginal features. For each leaf, we calculate the proportion of major features in blue and minor features in green. And then we can plot those and observe a really nice separation between different leaf shapes and margin types. So here the red circle contains all of the entire leaves, And the other 3 circles contain leaves that are either toothed, lobed, or toothed and lobed. We can visualize these proportions as species level distributions. So if we calculate the proportions for every single leaf in a species, you end up with 2 distributions, one for the major and one for the minor marginal features. And in this case, the little gray box on the left-hand side is a 5% threshold. If the mean of the distribution is inside that, it means that's an entire leaved species, which is what we see with this light blue Notholithocarpus species at the bottom. And in this case, the 4 gray species are some kind of non-entire leaf. So we repeat this, and we get lots and lots of plots that look something like this. We can normalize all of the species level plots from the previous slide and create genus level morphospace occupation. And we could even do that at the family level. So now that we've talked about measuring and categorizing leaves, we can discuss climate. So Earth has a finite set of possible temperatures and precipitation amounts. Depending on where you live in the world, it might rain every day or almost never. It could be above freezing most of the year or only for a few months. And a simple way to view sets of climate variables is through biomes. Well, there are many different definitions of a biome. For this chapter, I used the Whittaker definition, which simply relies on mean annual temperature, which is the x-axis, and mean annual precipitation, which is the y-axis. So species in the top right, in the green, are things that live in very warm, very wet places like the Amazon rainforest. As you go lower in the chart, you get drier, and as you go left, you get colder. For this chapter, I gathered climate data by mining GBIF for occurrence data and determined the median world clim bioclimatic variables for all of the species in our data set. So depending on the analysis, we either use the Whittaker biome definition or we use the 19 climatic variables. So as you can see in this plot, the species in a family tend to cluster in a few biomes, but this varies widely by taxa. We can look at the behavior of these 12 families by visualizing leaf shape within Whittaker biome space. So each of these plots is an individual family, and each pie chart represents the proportion of entire or non-entire leaves in each of those biomes. And right away, we can see that red tends to show up in the top right, indicating that entire leaves tend to show up in warmer, wetter locations. And we see blue generally tending to be in colder, drier locations. To really dig into these relationships, I performed quite a few different analyses, but today I'm going to highlight just the PGLS analysis that I ran for this. So on the left, we have a basic regression where essentially we're looking at all of the individual leaves in our data set. So in this case, this was performed with 860,000 leaves from oaks. And on the right, we have the species means. And we can look at the results in this overwhelming plot. We performed PGLS for every combination of leaf trait and climate. So while you take this all in, I'm going to take a sip of water. So let's break down what's happening here. On the left, we have the associations of leaf size with all of our climate variables. And on the right, we have the association of leaf shape with all of our climate variables. Everything on the left side of the black line is a temperature variable, and everything on the right side is a precipitation variable. Red boxes indicate a positive association, while blue indicates a negative association. And the strength of the color indicates the magnitude of the slope and the strength of the association between that leaf trait and that climate variable. There are 2 notable trends that I want to highlight today. One is that some families have stronger temperature associations, while other families have stronger precipitation associations. Here, the bold colors and the asterisks symbolize stronger, and in this case, a significant association. So for Nothofagaceae, it has a stronger association with temperature than it does with precipitation, and Cornaceae is the opposite. It has stronger precipitation associations. The second trend is that entire and non-entire leaved species within the same family often show opposite responses or lower magnitude responses to the same climate variables, like in Losaceae. And here we're splitting each family based on whether the species has predominantly entire or non-entire leaf margins. For Losaceae in particular, entire leaved species tend to have significantly stronger relationships with climate variables than the non-entire leaved species do, almost as if the non-entire leaves are moderating the influence of climate. So given all of these results, we can begin to answer our guiding questions. Do leaves get more or less complex in hotter environments? Well, for the family Moraceae, higher temperatures are associated with more complex leaves. But for the families Fagaceae and Vibranaceae, we see the opposite pattern. What about precipitation? The family Nothofagaceae, more of the complex leaves tend to be in higher precipitation areas. But for Urticaceae, we again see an opposite pattern. Do leaves get bigger or smaller at higher latitudes? This happens to be our first general trend that we found across all 12 families. Most taxa do tend to have larger leaves in warmer areas at lower latitudes. So the takeaway here is that phylogeny matters a lot. Many of the trends that I found are lineage specific rather than general rules, which is pretty cool. I want to show you a few more interesting plots, but to do that, we have to orient ourselves with this key first. On the left, we have a temperature arrow, and here it's a little strange because cold temperatures are at the top, warm temperatures are at the bottom, and that's sort of relating to latitude, where higher latitudes have colder temperatures, lower latitudes near the equator tend to be warmer. We have the same arrow for precipitation. We also have leaf outlines, which correspond to the different leaf margin types that we've discussed. So in this example, the most dissected and lobed leaves are the light green and light blue, and they tend to occur in the colder, drier regions, as opposed to the red leaves, which are entire, that tend to occur in the warmer and wetter places. The black dots signify leaf size. So in this case, the smallest leaves tend to occur at moderate temperature and precipitation levels, and the extremes tend to be the larger leaves. So to really visualize what's going on, this is the first of 3 major trends that we found in our 12 families. There's a lot going on here. The first thing to note is that the dark gray areas of the map are regions that experience temperatures below freezing in the coldest month of the year. The colors of the pies correspond to the leaf margin types in the key, and the size of the pie plot indicates the size of the leaf. Bigger pies are bigger leaves. So with Fagaceae, we can see that entire leaves, the red pie plots, tend to occur almost exclusively in warmer, wetter regions. And this is almost what we would expect with the marginality latitudinal gradient that we discussed, the Bailey and Sinnott work. But we find one more interesting pattern, which is that leaf area begins to increase again in lobed species at high latitudes, as you can see in the green pie plots in North America and the blue plots in Asia. And this is almost as if lobedness is enabling species to overcome thermal limitations that would otherwise constrain leaf size. With Cornaceae, we observe the influence of cold as a barrier, where very few species have been able to adapt to freezing conditions. And we also clearly see the leaf size latitudinal gradient. Most of the large leaves tend to occur in the tropics. Finally, with Urticaceae, we see a much more complicated pattern. With the previous 2 families, they're predominantly woody perennials. Urticaceae, though, it contains a lot of herbaceous taxa. So here we see 3 dominant patterns. We do see larger leaves in warmer climates. We do see a greater proportion of non-entire leaves in freezing areas. But we also see the influence of precipitation, particularly in Brazil and the United States. Entire leaved species tend to appear in drier areas of the Western United States, and the lobed species tend to occur in the drier regions of Brazil. So these 3 dominant patterns are just the tip of the iceberg. Large-scale analyses like this are important precisely because they can show us when there is not a general trend. So now we have the information needed to dig in deeper to better understand lineage-specific trends and define regimes like the three I just showed. So this macro scale data nicely shows that taxonomic rank and trait diversity are not equivalent across taxa. Some genera occupy significantly larger morphospaces than some families or even some orders. And critically, at this scale, we can identify areas in the tree of life with heritable variation, where the heritable variation is occurring. And sometimes there might not be an association between leaf traits and climate, specifically because there is not very much trait variation, which is itself an important finding. That brings us nearly to the end. Where do we go from here? The amount of data that I collected for chapter 4 could be used to answer endless questions. Even so, I already have data for most flowering plant families, and it's a mess. Let's just say that. So with my full data set, I have about 15 million leaves for 60,000 species. And plotting this data, we're truly in uncharted territory. I'm also working on ways to extract leaf vein architecture from these data, which I'll be working on as part of my postdoc with Selena Smith. And it's quite clear from chapter 4 that leaf vein architecture is an important piece of this puzzle that we just can't account for yet. And finally, with these data, we can statistically describe the morphological variation of most extant flowering plants, which is useful for potentially refining and revising paleoclimate models. And now all of the boxes are gray, which means that there's only one thing left. And I wouldn't be here without all of you. So thank you. I'm incredibly lucky to have ended up in this lab with Stephen and all of you. I've learned so much from all of you, and you've sacrificed a lot of your time to help me with crazy projects. So thank you. And now it's time for a little slideshow. I'm endlessly grateful to my parents who are watching on Zoom. They've supported every crazy idea I've pursued with enthusiasm and love, and I don't know where I would be without you, and I'll see you soon. So it takes a village to raise a grad student, and these are my people. I really don't know what to say, but all of you give my life meaning more than you could ever know. In my first year of grad school is when the pandemic happened, and I was very lucky to have a really fun group of pandemic pod pals. And I want to thank them for keeping my life vibrant and reminding me that there's life beyond research. Your friendship has been a lifeline. I also want to thank my cats, Sora and Ilya, for their quiet but loud presence during marathon coding sessions, for being the best co-pilots on the long drives to Colorado, and for reminding me that sometimes doing nothing is doing everything. And of course, the other furry animals in my life. So if you were sitting through this talk wondering what happened to Leaf Machine 1, well, that started 10 whole years ago, about the same time as my path leading me here. I was fortunate enough to be part of Stacy Smith's lab at the University of Colorado Boulder, and I was working with her then postdocs, Rob and Julian, and they were instrumental in teaching me how to be a scientist and helped me get my first author paper. So they gave me the freedom to explore and experiment, and I could not have asked for better mentors. And this is also when I met Shelley. We were both undergrad students trying to figure everything out. Shelley, you've been a great friend. Thank you for all of the conversations we've had about the chaos of academic life and for introducing me to all of the cool people at conferences. And finally, my favorite person, Sky. She's probably the only one more excited than me for all of this to be over with. Thank you for your unfailing patience and for listening to all of my rambling sessions and for offering wisdom when I needed it the most. And I can't wait for our next adventure. So thank you all for this.
Questions? Probably not the distribution piece, but.
That was really cool and very explained for understanding the computation and stuff. I noticed that you had like ideal leaves were being distinguished in Leaf Machine, and I'm wondering if you could also account for damage, like different types of damage that occur, because that might also be a trait that's interesting.
Yes. So that was a decision I had to make like 6 years ago, which is how to handle that. Um, in this circumstance, I'm defining an ideal leaf as a leaf where you can see the entire outline. So that is good and bad. It lets you study leaves, like if a leaf is being consumed by an insect, we can still find those with all of our algorithms. But on the flip side, for this kind of analysis, I do end up with a lot of messy data. So I'm trying to account for that by just measuring millions of leaves. So it does sort of come out in the wash. But there are thousands of leaves in my data set that you actually don't want measurements for. So that's also a next step in this process before we go too crazy with the 15 million leaves is filtering out some of the ones we don't want measurements.
Thanks for the one talk. So I'm curious about the link from the morphology here to ecosystem. You mentioned Sarah's talk saying that leaves are since it's extreme, we have lots of things measurements for the primary production. So I'm just curious about your thoughts. And maybe related to that, what's the sort of envision the response to climate change?
Right. I think that's a great question. So I think what I'm trying to get at is I think we really do need to account for phylogeny in all of this. So I do think all four of the hypotheses I presented for what drives leaf shape variation are all true to some extent and are probably more true for some taxa and less true for others. So I think part of it is, I know you work a lot with phenology, and the more we dig into it, the more it doesn't seem like there's one trend, like there's different trends for different taxa. So I think one of the missing pieces for me is the leaf venation data. I think that's a very understudied area. So I think I talk about this a little in my dissertation, but I kind of think that the shape is sort of a spandrel and that we really need to be focusing on how veins are growing in the leaves. Those seem to be more linked with the actual physiology of the plant, more so than just the shape that we perceive, which may or may not be directly relevant to the climate.
From one CU alone to another.
Oh, yeah.
Say CU voltage, UC voltage. I do have a question. What actual step getting the digitized label information to check if this person have to sit there and read both of them? What
So that is a continually evolving process right now. A year ago, it was a lot more complicated. Now, if you have a typed label, it basically is perfect. If you have a handwritten label, we still have to do quality control. There's a lot of ways people are figuring out how to deal with that. You could use multiple large language models, multiple methods, and try to do sort of like an alignment of the text. So you could at least get like a confidence score for it. So that's one approach. I've been trying to build like a gold standard data set so we can actually have a data set to act as sort of a benchmark for transcription. The reason we want to integrate directly with the data portals is because that's where the data is stored already. So internally, we have an editing system that lets us sort of efficiently go through all of the corrections. But it would be a lot better if as soon as an institution uploads the image, as soon as they take it, they get a preliminary text response, and then we can edit that at any point. So part of the problem is a difference between something that could be useful internally to the institution. You would want the data right now, even if it's a little messy. It could, and then at that point, we could justify taking 10 more years to get through all million specimens. So it's kind of just a trade-off of accuracy versus expediency.
All right. Thanks. Let's get one more value thought.
So yeah, so now the dissertation committee will be in here, and if everybody else can exit and maybe you want to hang out by the kitchen or something, you can we presumably celebrate. Great job. Yeah. Bowls of edible leech.